Hello, welcome. My name is Bruce Bryan, and I am really excited to welcome you here today, both on behalf of miniPCR Bio and our collaborators at the Duke Lemur Center. Today, we're going to present our Conservation Genetics Lab, Identifying Lemur Diversity. Before we really get into this, I just want to note a couple of things. We're presenting this today both for teachers and students. In the description below, there's some links there. You can go, there's a worksheet with questions to follow along if you'd like to do that. There's some other links there. This PowerPoint also will be available afterwards. We'll paste it there. And if you would like to download it, you can. It's not loaded yet, but it will be soon. And then finally, this webinar will remain on this channel after we present this today. So if you'd like to come back and share it later, you can. So again, welcome. I'm here at my home in Rhode Island, but today we are going to take you on a really interesting adventure to Madagascar, and I'm really excited to do it. So our goals for today, we are going to look at lemurs. Specifically, we're going to be focusing on the dwarf lemurs, and we're going to use them as a model for both conservation and evolution. And these things today, evolution and conservation, fit really well together because both of them use the same modern genetics tools. And we're going to use those tools to gather evidence. And using inference from evolution, we're going to track species relationships. And once we know those relationships, we're going to use that to inform decisions about conservation. If you're not familiar with miniPCR Bio, we make biotechnology equipment. Specifically, we make small, robust, affordable equipment that finds a home most widely in classrooms around the country and around the world. And as part of that, we consider it part of our mission to provide innovative curriculum that really drives science for students and provides them authentic science experiments and learning experience. The other place our equipment finds a home, however, is with field researchers. There's a lot of folks today who are now taking mobile biotechnology equipment like ours to the field to directly answer questions at their site of research. And so today we're excited because we're gonna be able to marry those two things. We're going to present to you research that was done in the field, some of it with our equipment, but we're going to present it in a way that you can use our equipment to teach students. So this is our equipment in the field. This is it being used this comes from a paper that was published just in August of 2020. These are our collaborators that I'll introduce you to in just a second. So they took ours equipment to the field to study mouse lemurs. And just as I said, they used the tools of modern genetics to answer questions directly at the site of their research in Madagascar. But then they also took these same tools in Madagascar around to do outreach with local communities, schools, and universities, training local scientists on the use of genetics and the genetics of lemurs. And so when we saw this, it really is such a perfect marriage of what they do and what we do, taking tools to the field and also in using them for outreach. So we worked together to develop an activity that they could both tell the story of their research and use in their own outreach activities. And then we can tell their story of research and have students do those same activities in schools. I'll just note I've cited the article right here, and that article is also linked in the description below. If you're interested in reading a science article in a high school class, it's really a great one. It's open access, so it's free to download, but it's also relatively short and it's very accessible. So with that, I'd like to introduce our collaborators from the Duke Lemur Center, Dr. Lydia Green and Dr. Marina Blanco. I could tell you all about Madagascar and tell you all about them, and I will be doing that some later on, but I think it's best if they introduce themselves. Before we do that, though, I'll just say Lydia and Marina have joined us today in the chat. So if you have any questions as we go along, pop them in the chat and you'll have two certified lemur experts there to answer them. They're both using the Lydia Green username though. So they're both under that one username. So with that, I will let Lydia and Marina introduce themselves and take it away.
Hi everyone, I'm Lydia Green.
And I am Marina Blanco.
And we're research scientists here at the Duke Lemur Center in Durham, North Carolina. And we're also two members of a large team that's fascinated by this question of figuring out exactly which lemur species live in which forests in Madagascar. And so for today's conservation genetics lab, we're going to be talking about the incredible dwarf lemurs, some of our favorite animals that live in the Sindjarivu forest. And that's Sindjarivu.
But before we get started, I think we should answer some questions. What is the Duke Lemur Center?
So the Duke Lemur Center maintains the largest and most diverse collection of lemurs outside of their native Madagascar, and we're a center for lemur conservation, education, and research, all of our research being non-invasive and non-harmful to the animals, of course. And if you want to learn more about what we do, you can check us out at lemur.duke.edu. Can you tell us a bit about how you got involved in lemur science to begin with?
It was a little bit by chance. I was studying anthropology in Argentina, and then I became interested in evolution, trying to figure out why different animal species look different. And I didn't know at the time that I was going to become really passionate about lemurs in general, and especially mouse and dwarf lemurs, who happen to be cryptic, meaning that there are many different species, but pretty much all of them look alike. More of that later. Next question for you. So what lemurs do you study?
I tend not to study so much the tiny nocturnal mouse and dwarf lemurs, although I do when we're working on these projects. But my own research tends to be more focused on these guys, the shifox, and the sort of big diurnal, day-living, flashy, very visually distinctive species who are just incredible. Do you have a favorite lemur here that lives at the Duke Lemur Center?
Yes, I do. And of course, it's a dwarf lemur, and her name is Sandpiper.
Sandpiper.
And so what about yours?
Gertrude. She's just fabulous. Can you tell us a bit about why our team started working at Sindjarivu in the first place?
So we went to Sindjarivu back in 2006. Actually, the story begins a little bit earlier, 2 years earlier in 2004. That was when I first traveled to Madagascar, going to Ranamafana National Park. Ranamafana National Park is a mountain rainforest in southeast Madagascar. I was looking for mouse lemurs, but quickly became intrigued by their larger and more elusive cousins, the dwarf lemurs. Though larger, these lemurs were much harder to see, especially during the dry season. And I quickly learned that's because they were hibernating. As my passion for dwarf lemurs grew, I also began working with them at different sites. Sindjarivu sounded both appealing and intimidating. Our team would have to face serious challenges to study dwarf lemurs there. We had to set traps high up in the trees. We had to travel several miles every day up and down hills to find the lemurs' sleeping locations. And we had to navigate the forest through rain, mud, vines all night long. It became clear from the beginning that success depended on the strengths of teamwork. The longer we worked at Sindjarivu, the more questions we had. We noticed some differences in size, length, cross-legged. We might have also stumbled upon a population of sea-raised dwarf lemurs, a lemur long thought to be extinct, but who likely once lived at Sindjarivu.
And so this isn't the end of our story at Sindjarivu, but it is the beginning of yours, because for today's Conservation Genetics Lab, you're going to pick up with the next phase of our work as we try to determine which dwarf lemurs actually inhabit Sindjarivu today. And so you'll be figuring that out through the lab today. So good luck, and hopefully you come up with the same answer that we did.
All right. So that's where we're going to start up again. Have researchers discovered a thought to be extinct species of lemur? I mean, this is really exciting. There's not too many finding new species that goes on today. There's a lot of extinction that we hear about, but today we're going to ask, have we found a new species? And we're going to again be using the tools of genetics and evolutionary biology to inform important questions surrounding biodiversity and conservation. And we're going to do that by looking at actual data and looking at the actual lemurs that were used in this study originally. So before we do that, we want to step back and just talk about Madagascar for a second to give us a little bit of context. So this is Madagascar. It's here off the coast of Africa. And Madagascar is an island. It's a large island. In fact, I believe it's the world's fourth largest island. It's also been an island for a very, very long time. Um, so long that the animals that live on Madagascar, the plants and animals that live on Madagascar, are completely unique to there. And that's because they've been evolving on Madagascar, unconnected from other land masses for tens of millions of years. And when I say Madagascar is large, it's big. It's almost twice the size of Great Britain. It's somewhere smack dab in the middle of between the size of California and Texas. So this is a huge landmass. So lemurs got here. Well, we don't really know how they got here, but somehow they got here across 250 miles of ocean. That lemur ancestor that made that voyage looked something probably like this. We don't know for sure, but this is an artist's rendition of a fossilized primate that was alive about the same time and probably closely related. Most likely what happened was they were swept out to sea in a storm, holding on to a tree or a mat of vegetation. A small family of lemurs or small group of lemurs washed out to sea and then washed across 250 miles of ocean until they landed on this island. And this large island was virtually devoid of large animals, large mammals. And so the lemurs that entered there entered a habitat that was just open ecological niches that they could take advantage of. In Madagascar, you have a whole bunch of habitats here. You have in the south, it's dry and arid. We have rainforests all along the east coast, deciduous forests along the west, and in the center of the island, you have the high central plateau. And so as lemurs spread out across the island, they encountered different habitats. And as they encountered different habitats, the pressures of natural selection changed them, and they evolved into the many different species that we see today. Today, there's over 100 species of dwarf lemurs, from the small mouse lemur here, which is literally the size of a mouse, about 3 inches long and weighing just an ounce, to the large indri, which is about a little over 2 feet long, 20 pounds or so, so about the size of a mid-sized smallish dog. And then, of course, we can't forget Zubumafu here in the middle. So lemurs are primates. That means in the Linnaean hierarchy, right? Kingdom, phylum, class, order. We are the order primates, and that's the same order that humans, apes, monkeys are. Though lemurs are as distantly related to humans as you can be and still be in the same order. Being primates, it means that they share some things in common with us. We have grasping hands with fingernails instead of claws, though lemurs, like most other primates, also have grasping feet. All primates have binocular vision, so 2 eyes sort of centered in the front of the head, and that gives us really excellent depth perception. And that sort of goes along with the fact that most primates, and certainly primate ancestors, are arboreal. They live in trees. If you're jumping from branch to branch, it's really important to have good depth perception. As I said, today, there's about 100 different species of lemurs alive in Madagascar. And sort of interestingly, that number has been growing as new lemur species have been discovered, and that's going to be a major theme of this lab. But at the same time, it's also shrinking because these lemurs are under a constant threat of extinction due to habitat destruction. And then my just like favorite little note here, proving that the coolest animals are the extinct ones. The largest ever lemur was about 350 pounds. So you have to imagine something that looks sort of like this, but was the size of an adult male gorilla. So as I said, today these lemurs are under the threat of extinction due to habitat change. Many lemurs are. And the habitat on Madagascar has been changing for a while, and most significantly, recently, it's been changing due to human use. Humans first colonized Madagascar in sort of a large, grand sense with large civilizations only about 2,500 years ago, which is really recent in human history. And as soon as they got there, just as humans entering any landscape will, they changed the habitat and put pressures on the animals that live there, whether it be from hunting or just cutting trees and farming. Today, lemurs are most under threat from loss of the forests. It's since about 1975, more than 33%, more than a third of the forests on Madagascar have been lost. And that's largely due to the effects of agriculture. So here we see, the forests on the top of that hill, and folks have had some pretty intensive agriculture on the side of it where no lemurs can live there now. Even places where folks have farmed but now left fallow, it can take decades for those forests to recover if they ever do. And then even in places where there are still intact forests, the trees in those forests can be valuable, and specific species of trees, especially the largest, can be quite valuable, and people can go in to take the wood. And when people are selectively harvesting specific species of trees, and specifically the oldest trees, that changes the habitat in ways that can affect different species differently. And while we're not going to talk about it a lot today, I just do want to note that layered on top of all of this is the effect of climate change. So as lemurs' forests are shrinking and they're becoming more fragmented, the forests themselves are changing due to the effects of climate. My slides are frozen, 1 second. All right. There we go. Sorry about that. So let's introduce you to the lemurs that we're talking about today. This is Carigalius major, the greater dwarf lemur. The greater dwarf lemur was once thought to be one of only 2 species of lemur that lived on Madagascar. The greater dwarf lemur lived across the east coast here, while along the west coast there was a different species. And it was this is the range that people thought was this one species, and they thought it was a widespread, very healthy species. But genetic technology has now told us that what we thought was one large, widespread species is actually many. Today, instead of those 2, there's at least 8 different species of dwarf lemurs that have been found. So we're actually not going to talk much about the greater dwarf lemur today. Today's story focuses on this guy, the furry-eared dwarf lemur, or Carigalius crossley. So you can see this lemur lives in just one, or actually 2 sort of portions of that, what they thought was the original range of that one big species. Because as scientists went in, Marina being one of them, and looking at the lemurs, they realized that there were differences between them. And the question was, is this variation within a species that just in different places it looks a little different, or are they actual different species? And the answer is they're different species. Today, C. crossley is considered vulnerable by the IUCN, so it's not quite endangered, but it's certainly not as healthy as the species that they thought it once was if it covered that entire range. Now, one of the largest populations of C. crossley lives at Sindjarivo, which is the forest that Lydia and Marina were telling you about, and Sindjarivo is right about in here on Madagascar. Now, we've been mentioning species a lot, so I just want to say for a second, we should identify what a species is, and we do just need to note that this is a bit of a danger zone. It's always good to go back to some foundational work. So the origin of species, you'd think Charles Darwin, he writes a book about species, he would have a good definition for it. But this is just a little quote I like from the book. This is how he defines a species. In determining whether a form should be ranked as a species or variety, the opinion of naturalists having sound judgment and wide experience seems the only guide to follow. So even Darwin, in writing Origin of the Species, just sort of throws up his hands and says, yeah, I'm just going to trust that people know what a species is, but we're not going to actually define it. Defining species has been a source of big argument in academia. And really, there's a lot of different species concepts that you could have. If you're dealing with bacteria, if you're dealing with oak trees, if you're dealing with lemurs, you may have to use different species concepts. You may have to use a different species concept if you are a paleontologist versus whether you're a conservation biologist. So today, we're just going to throw down what our species is. Our definition is going to be, well, we're dealing with animals, so it's a group of animals that can regularly reproduce together in the wild, that shares a particular set of characteristics and evolutionary history. So to me, the most important thing to take away from that is most species concepts have something to do about reproducing together. So this is a group that reproduces together regularly. And that idea of evolutionary history, not only do they reproduce together regularly, but they have been reproducing together with each other and not with any other animals for a very, very long time. So to think about this a little more, we are going to just look at a little vignette here. And my PowerPoints have frozen on me just a little bit. 1 second. There we go. So cryptic species. Species that are difficult or impossible to tell apart from morphology alone. And that's what Marina alluded to, that some of these lemurs that we're talking about, that they are cryptic. So these are African elephants. On the left, we have African elephants, and on the right, we have African elephants. It used to be that folks thought this was one species all across Africa, that there was the forest elephants that live sort of more on the west side of Africa, that there were the African bush elephants that live more in the savannah, but really it was just one continuous population, and they bred together, and there was no distinctive line between them. And this was the idea until just recently, when people did genetic studies on them, and they learned that not only were these things not reproducing with each other today, but it looks like these two populations haven't mated with each other for any significant amount of time, or for any significant amount for almost 2 million years. Okay, so for 2 million years, these have been different populations that haven't been reproducing together. So we definitely consider these different species. And that's just to also know other species contain a lot of variation. So this is a picture of a pack of wolves. And this one pack of wolves probably contains more variation within it than we may see between different species of lemurs. So again, it's one pack. That means they're all very closely related. It's parent, offspring, siblings going on here. And yet there's a lot of variation. So the take-home message from that little story is that morphology is really important. I don't want to discount morphology. We are going to study it today. We're going to look into it, and it's really important to understand the morphology of organisms. But when you're making species classifications, when you're trying to understand how things are actually related to each other, the genetics is going to rule in the end. The genetics is going to tell us how these things are related and make our final species classifications. So, the furry-eared dwarf lemur. When Marina and other biologists went into the forest at Singirivo, it seemed like the furry-eared dwarf lemurs that were living there, that there was more variation than you would expect to see in a single species. And so the question was instantly, is this another species of lemur? Or is it something where there's just a population that has a lot more variation than you would typically expect in one of these species of lemurs? The twist on this is that the lemurs that didn't look quite right, they looked like this other species of lemur called C. cibriae, or Sibrey's dwarf lemur. Now this is really interesting because this lemur was thought to be extinct. The last known population was lost when the forest they lived in was cut down. Today, it is known only from a single museum specimen, and no genetic evidence is allowed to be taken from that specimen because of regulations. So we know nothing about this species' genetics. The sort of odd lemurs at Sindrivo seem to be resembling this lost species. So that's what we want to find out today. What are the lemurs that we're looking at? To do that, we're going to do 3 bits of analysis. We're going to do a morphological analysis where we actually look at the lemurs. We're going to run a gel and do a preliminary PCR RFLP analysis. That's the sort of hands-on part of the lab. And then finally, we're going to do sequence analysis to really establish what species relationships are. So first, let's meet the lemurs. So these are the 8 lemurs that we're going to talk about in this study. And I would just say, if I was teaching in this class, I think the first thing I would do would be to have a class poll to see who is the class mascot, who our favorite lemur is. So I'll suggest, if people are interested, you might just throw down in your chat, in the chat there, who you think the best of these lemurs are. I would vote for Garafa. These 8 lemurs come from the original study that was performed, and we're going to look at them and the actual data that was taken on these 8 lemurs individually to make our conclusions. So to do that, again, we're going to start with a morphological analysis, but first, because we're doing things a little bit cooking show style here, we're going to actually run the gel first. So we're going to start with the gel. I realize you don't know why we're running the gel or what we're going to look at. That's okay. By the time we look at the gel, we'll know about it. Okay, so we're going to start by loading the gel, and then we will jump back to the beginning and work our way all the way through the workflow. All right, let's load and run the gel. So this is my blue gel set up here. And I'm going to start by just adding a DNA ladder to my wells. Again, we're going to talk about exactly how all of this works when we get there in just a little bit. There we go. So, and then I'm going to go through, and we have all 8 samples here that we're going to load one at a time. In the actual lab, students could load all 8 samples, or there is a way in the lab written in that students can sort of make a prediction ahead of time and only load a subset of those samples if depending on how many or how much your gel allows you to run in the classroom. So as I load the samples, this is an agarose gel, and I'm putting the DNA up here in these wells. And I use my pipette tip. I just put it sort of right over the well, and I slowly press down. And because there's a dye in there, the DNA falls down into the well. And then what we're going to do is we're going to turn this electrophoresis system on, and it's going to separate all the DNA that I'm putting in here by size. It's going to use charge to pull things through the gel, and they will separate by size. For the activity that you would do in class, this is the hands-on portion of the lab. This DNA is provided to you, and you would just distribute it among your students. You'd run a gel, and you would do the analysis of the gel once it's all run. And again, you don't know why I'm doing this right now, but I promise you by the time we look at the results, you will. Last thing I just want to say is this is the blue gel electrophoresis system that we make at miniPCR. This is one of the tools that a lot of folks are now taking to the field because it's an entire electrophoresis and illumination system built into one. And so it's everything you need to run a gel, and it's probably much smaller and simpler to operate than other gels that you are familiar with. All right, so I've loaded everything up there, and I'm just going to put my lid on, press the power button. As long as that green light is on, we know it's running. It's going to take us about, well, we'll be able to see results in about 10 minutes. We'll get nice results in 20 minutes. In class, I would try and run it for about 25 minutes to get some really nice results. Oh, all right. So let's jump backwards. So next we're going to do our morphological analysis. So morphology, sometimes called morphometrics, is just looking at form and structure. So we're just going to measure the lemurs, see how big they are, see what color they are, measure different parts of them. And that's to, just see how things are similar to each other or how they are different physically. So to do this, in the field, lemurs are captured in traps and brought back to the field station where they're measured. A sample of DNA is taken, and then very quickly, as quickly as possible, the lemurs are returned to where they were captured. Sometimes they're outfitted with a little radio collar so that they can do other studies with them. And this here is Marina doing just that. So to do the morphology or the morphometrics, unfortunately, we can't mail you lemurs to measure, though that would be especially fun. So what we've come up with, we have these sort of lemur trading cards, and as part of the lab, students will cut these out, and you'll have one for each lemur, and we'll ask students to analyze the data that's there and try to put them in groups. So you can imagine students sitting around a lab bench with 8 of these cards, looking at all the different information here and deciding, is this one group, is this 2 groups, how do we split these up? The data that you're going to see here starts out with just some basic stuff: the sex, female or male, age, they could be juvenile, adult, or subadult, when they were captured, and that capture time was that pre-hibernation or post-hibernation. Dwarf lemurs are obligate hibernators, which means every single dwarf lemur hibernates for several months during the year. They hibernate during the Madagascar winter, which is our summer. So in March, that would be pre-hibernation, and then I think the other ones are caught in October, which would be after they hibernated. Now that information that I just told you about isn't really that important to putting them in groups, but it may give you a clue, or it may let you think about the other data that you're collecting and how much you should weight it in different ways. So for example, we look at coat color. That's just sort of the back of the lemur. What color is it? Alicia here is reddish brown, Rudolfo's silvery gray. Face color, you can see that Alicia has a little bit of white on her face, where Rudolfo's fur just sort of fills in around the eyes. All the lemurs have these black rings around their eyes. Some of them have a white tail tip. Alicia does, Rudolfo doesn't. Some have a dorsal line. That means a black stripe going down their back. Rudolfo does, Alicia doesn't. We measure their face shape. So one of the things that folks have keyed in on these lemurs, some of them seem to have a bit of a pointy snout like this, and some of them, their snout is a little bit more rounded. And then finally, we take measurements. How long is their head? How long is their tail? And how much do they weigh? So that's the data that students go through, and they go through it for all 8 lemurs. We have this all out, and you can imagine, sitting around saying, I think we should base it on this, I think we should base it on that. That's very difficult to do in a virtual format, and so we are just going to present you some hypotheses, and this is built into the lab as some sort of scaffolding. So if students are having trouble, you can present these ideas to them. So Marina, we say her hypothesis is that we should break these lemurs up by size. Historically, the furry-eared dwarf lemur is thought to be a little bit bigger than the C. cibriae. So what if we break them up into 2 groups by size? We would have our 3 biggest lemurs over here, Mbola, Razafi, and Alicia, and our 5 smaller ones. So that's one possible hypothesis. And if you want, in the chat, you could throw down, does size, again, historically, one species is thought to be a little bit bigger than the other. So does that seem like it might be a good idea on how to break them up? What are some advantages to breaking them up that way? What might be some problems with breaking them up that way? Next, we have Jean Basile's hypothesis. Jean Basile is one of the local scientists on the team. So Jean Basile notices that there seems to be two basic coat colors going on. There's a reddish brown coat color and a silvery gray coat color. And so if we break things up that way, it looks like this. We have 5 of these possible furry-eared dwarf lemur, C. crossley, and 3 C. cibriae. Again, if you want to throw in the chat why you think why you think that may be a good way to break them up that way, or maybe why there may be some drawbacks to that. And finally, Jules, another one of the local scientists on the team, Jules suggests no, this is just a species with a little bit more variation. He thinks the variation that they're seeing is within species norms, so maybe we just think there's one species. So we make predictions using that morphological analysis, and then we're going to do our PCR RFLP. And doing this would generally be considered a bit of a preliminary analysis, but this is the way that we're going to tell these things apart in the classroom. So as we said, the best evidence for testing species comes from genetics. So then the question comes, what genetics do you use? Do you just sequence the entire organism? Well, that's way more data and work than you actually need. So we're going to take a shortcut and we're just going to look at one gene. The gene we're going to look at is cytochrome B, or sometimes called CytB. CytB is a mitochondrial gene, and we use it for a few reasons. One reason is just that folks have used CytB to break up species for a long time. So it's a 1140 base pair gene located in the mitochondrial genome. And just real quick basic biology to get everyone up to speed. You have your nucleus in your cell, which contains your nuclear genome. When you're talking about like your genome or an organism's genome, you're usually talking about its nuclear DNA. That's the vast majority of DNA and variation in DNA. Everything that we're talking about is their nuclear DNA. But then cells also have mitochondria, these organelles that are out in the cytoplasm that convert energy into a usable form for the cell. And they have their own genome. It's a very small circular genome that just contains a handful, 13 genes. We're keying in on one of them, the sequence of CytB. And we use CytB, or we use mitochondrial genes, and CytB is one of those mitochondrial genes. We use mitochondrial genes because they evolve at this Goldilocks rate, where it tends to be that there's enough differences between closely related species that you can tell them apart. Okay, so there needs to be enough differences that when I look at the DNA, I can say, yes, this is one kind and this is another kind. They're definitely different. But they don't evolve so fast so that if you're looking within a species, the sequences tend to be very similar. So not only can I look at 2 and say, yep, this one's different from this one, I can take another one and say, yeah, these 2 go together. These 2 are definitely the same species. We are going to do what's called a PCR RFLP analysis. PCR RFLP stands for polymerase chain reaction restriction fragment length polymorphism. That's a big mouthful, which is why we abbreviate it. The idea is this. We're looking at the 1140 base pairs of the CytB gene, and we have previously found a polymorphism, a difference, at base pair 682. And at that base pair, there's a unique genotype, a set of letters that is only found in C. crossley, the furry-eared dwarf lemur. Every other lemur that has ever been sequenced has a different set of nucleotides at that base pair 682. The sequence in C. crossley just happens to match what's called a BGL2 or BAGL2 restriction enzyme cut site. So the first part of that word, PCR, the first thing that we need to do to analyze the DNA is we want to key in on that 1140 base pairs. PCR is is a mechanism for finding a very specific region of DNA in a complex sample and then making 1 billion copies of it very quickly. And obviously from the name of our company, that's something that we do a lot of. We have a lot of labs that focus on PCR. In this lab, you don't actually do the PCR, but it is something that you could learn about as part of the lab. So again, PCR, the first step is to take the complex sample of DNA, that sample from the lemur, key in on that 1140 base pairs, and then make 1 billion copies, just so all we're looking at is just that section of DNA. We're then going to use that Bagel2 enzyme to key in on the sequence that we want. And Bagel2 is a restriction enzyme, which means that when you add it to DNA, it recognizes a very specific sequence. And when it comes in contact with that sequence, it cuts the DNA. Bagel2 specifically recognizes the sequence AGATCT. Anytime that Bagel2 is mixed with DNA and that sequence comes up, it's going to cut the DNA. So here's a sequence of DNA. We go along, there it is, AGATCT. Bagel2 will recognize that. The enzyme will come in and cut it, and now instead of 1 piece of DNA, you have 2. So in this experiment, it means that once we get that site B gene, and we're actually going to deal with 1,200 base pairs because we're dealing with just a little bit on either side of the site B gene as well. We mix it with that Bagel2 enzyme, and in the C. crossley, the furry-eared dwarf lemur, it's going to find that sequence, it's going to cut it, and if it's a furry-eared dwarf lemur, that site B gene, that site B sequence of DNA will be cut in 2. Any other dwarf lemur, that sequence won't be there. So when you mix it with a restriction enzyme, the enzyme just can't cut and nothing happens. I think my PowerPoint's frozen. One more second, we'll wait for it. I apologize. One thing to note here though, while we wait for my PowerPoint to catch up, we're comparing this C. crossley or any other dwarf lemur. So we aren't keying in on C. cibriae yet, and one of the reasons is we don't know the genetics of C. cibriae. So we have to make the assumption that if this is not C. crossley, because of the morphological analysis that we're going to be making, we're going to just have to assume that C. cibriae is the other species. Okay, so this is still a little frozen. That's all right. So again, the idea here, and we can already start to look at our gel. The idea is that once we key in on the species, if in our gel we see 2 bands of DNA, we will know that that DNA came from a furry-eared dwarf lemur. If it has come from any other dwarf lemur, we will know that it will only show 1 band of DNA. So I've got my gel here, and I'm going to switch over. And all right, we've just been running the gel for a few minutes, but we can already see our results. If we go along, we can see, we can't quite see what's going on here yet, but you can clearly see that some of these bands are different. And if we were to get nice and close to it, we'd be able to see some clear results. So my PowerPoint's back up and working. Again, sorry for technical difficulties. So let's look at this again. The idea here is that our Bagel2 cut site, if it cuts, we get, we believe for sure that is C. crossley. If it does not cut, we believe that it is a different species of lemur, and based on the morphology, we think that's C. cibriae. So I've already shown you the gel a little bit, but the idea what happened in that gel is that we put DNA in the wells up here at the top of the gel, and we applied an electric charge to that gel. And so the DNA in here was pulled down through the gel. This is what a gel looks like on the microscopic level. So it looks like this complex web that the DNA has to get pulled through. And you can imagine that smaller pieces can get pulled through this much faster than larger pieces. So as we run our gel, in lane 1, in our first lane, we put a DNA ladder. And that DNA ladder gives us known size standards. So we put in DNA that was 100, 200, 300, 500, 700, and 1200 base pairs long. As this gets pulled down, if the DNA got cut, we'll see 2 bands at 500 and 700. And if it doesn't get cut, we'll only see that 1 band at 1200. So we already looked at the gel. Again, if I were to look closely, I can see the differences already. It's only been a few minutes here, but instead I'm just going to show you it running. This is a gel that I ran yesterday over about a half an hour. It's going to run here in 15 seconds. So again, this is something you can definitely run in a class time. This is run in half an hour. I could see results within 10 minutes. I could see results clearly in 20 minutes. All right, so I loaded these individuals into the gel. I loaded them in alphabetical order. So going along, we have, well, I guess in the first lane, we have 1, 2, 3, 5, 7, and 1200 base pairs. And then we can look at our individuals. Alicia, Bima, Garafa, Ihari, Malala, Mbola, Razafi, and Rudolfo. And again, reminder, 2 bands means that it's we know that 100% means that it's a C. crossley, the furry-eared dwarf lemur. And 1 band, we believe, well, we know it's another species of lemur. And from the morphology, we're going to assume that it's the Sibrey's dwarf lemur. So this is our grouping. We know we have 2 groups. The groups happen to look like the hypothesis from Jean Basile. And here it is. So we believe we have C. crossley and C. sibryi. Just to take the point home, guys, we rediscovered a species. There's I don't know, there's not a lot of good news in the world today. This is pretty exciting. Rediscovering a species, and your students can do this by looking at the morphological data, looking at the genetic data, and looking at actual data from the field that scientists use to make the same conclusions. It's not the end of the story, though. Habitat change continues on Madagascar. So the forests of Sindjarivo, like other forests on Madagascar, are under threat. And they are under threat from encroachment, mostly from agriculture, but also from climate change. And my PowerPoint's hanging up on me for 1 second again. I'm working on it though. And so we want to do a little bit more analysis and think about, now that we have rediscovered this species, we want to gain an insight into the health of the different species. And now that we know that these species are different species, how are they responding differently to habitat change? All right. So to do that, we are going to look at some data here. Whoop, sorry, I clicked through a bunch. We're going to analyze trap data. And so these are the 2 species. And this is, again, actual data from the field. I'll just note that the study that this is all based on was published in 2010, but the data goes back to 2006. That's partly because Marina and the other researchers there have a pretty keen eye, and they knew what they were looking for early on. So we're going to analyze something called trap night data. What a trap night is if you put a trap out in the field and you check it the next morning, 1 trap for 1 night, and you check it the next day, that's 1 trap night. If you have the same trap out for 5 nights and you check it every morning, that's 5 trap nights. If you have 5 traps out in the field and you check every one 5 mornings in a row, that's 25 trap nights. And so what we're looking at is trap nights and the total number of lemurs that were trapped per trap night. and that is a proxy for how widespread the animals are. If you're catching a lot of lemurs really easily, it's probably because there's a lot of traps out there. I'm sorry, it's probably because there's a lot of lemurs out there getting caught in the traps. If you're not catching them very easily, it's probably because there are fewer lemurs out there. Before we look at this data, I just want to point out that in some of these years, we're talking about 900 trap nights. And if you just remember, Marina talking about hiking the miles through the rain to get to find the lemurs and the traps up in the trees. This is 900 trees climbed to look in a trap, and being successful like less than 20 times finding the thing that you found. It's a phenomenal amount of work that I'm quite impressed by. So let's graph it out, and this is data that is presented for students and for them to analyze. So on the left hand, on the y-axis here, we have the percent success. So what percent of lemurs, or what percent of trap nights did you actually find a lemur in the trap? And then we have years, and this is going to go from 2006 to 2019. So early on, they were catching between 7 and 7.5% success rate, but then you can see it just dives right down. From 2011 till 2019, it's about around 2% or lower success rate. That's really concerning, right? Not only is the furry-eared dwarf lemur, we know that's vulnerable. We've just found a species that we thought was extinct, so it's got to be, extremely vulnerable itself. And every year they're going back, it's harder and harder to catch these lemurs, which means their numbers are in serious concern. We also have data that students can analyze based on the percent of species that were caught each year. So on this axis, it's just of those successful catches, what percentage was one species and what percentage was the other. So the first year that they caught lemurs, they actually caught only Sibrey's dwarf lemur, and they didn't catch any of the furry-eared dwarf lemurs back in 2006. Now, if we watch this data go across, and just know this data is sort of inherently a mirror image because the two things will always add up to 100. But if we watch this advance over time, right, you can see another pretty disturbing trend. The Sibrey's dwarf lemur that we just rediscovered, thought it was extinct, but its numbers seem to be at least very worrying. Every year it looks like it's getting harder and harder to catch these lemurs, where the furry-eared dwarf lemurs are relatively more numerous, at least compared to the Sibrey's dwarf lemur. So this is very concerning, and this is going to inform conservation decisions for sure. We give you one more bit of data to analyze. We're not going to go through it now, just in the interest of time. But the story here that sort of ends up being told is that this is a story of generalists versus specialists. And if you teach environmental science or AP environmental, you know that this is a key concept. The furry-eared dwarf lemur, C. crossley, is more of a generalist. It has a wider diet, and it's much less picky about where it makes its nest. It makes nests in trees, but it'll sleep in all sorts of different places. C. sibreye has a much more narrow range of diet, and it also is really particular where it makes its nest. It will only make its nest in tree holes, and that can be problematic, especially when we talked about at the beginning, if certain trees from the forest are being removed selectively, especially the bigger, older trees that are more likely to have holes in them. That could be really problematic for C. sibreye. So we took a little detour there. I just want to finish to talk about the conservation aspect. I just want to finish by doing a little bit of sequence analysis. And this is the real evolutionary part of this. So the last thing that we do in this lab is that we give you the DNA sequences from those 8 lemurs that you collected. These are, again, the DNA sequences that were collected in the original study, and they are linked to the lemurs in the lab. We also give you 3 DNA sequences from C. medius, the fat-tailed dwarf lemur. That's the dwarf lemur that lives mostly on the west side of the island. We also give you 3 sequences from C major, the greater dwarf lemur that I mentioned at the beginning of this story, another species that lives on the east side of the island. And then finally, we give you the sequence of Microcebus murinus, the grayed mouse lemur. This is one that we know is not related to these guys very closely at all, and it's in there for reference and also sometimes for technical reasons. So we present these to you in just a text file and ask you to analyze them. And we'll ask you to analyze them using the tools of bioinformatics and evolutionary biology. And we'll walk you through that in just 1 second. Before we do that, though, we want to talk about so we're going to use this. My PowerPoint's frozen one more time. We're going to wait for it, but I have plenty to say. Don't worry. We're going to build an evolutionary tree with this. And so using those evolutionary relationships, we're going to really confirm that indeed this group that we found, the C. cibrae, is indeed its own species and not related to any other species directly. Well, it's going to be related to them, but not perfectly related to them. It's going to be its own thing. But we want to understand how things are related to each other. So we're going to build what's called a cladogram or an evolutionary tree. And so we have this example one here that I'm just going to talk you through. So again, this is a cladogram. We are looking at time here going in this direction. So in any cladogram, you put your current or most recent animals at the end of this tree. These are called the branches of the tree, and the animals that you use to build it go on the end. And then as we go back in time, in this tree we move right to left, going back in time. But these trees can be oriented different ways. Sometimes you'll see them going from top to bottom, bottom to top. Sometimes they're in a circle. Sometimes they're slanty lines. Sometimes they're right angles, like in this one. It doesn't matter. What we're really concerned about looking at this tree are 2 things. We're going to follow branches like this back, and we're going to see where they meet at things called nodes. So in a tree like this, if I want to know who is the lion most closely related to, I'm going to follow its branch back until I reach at the first node, the point where 2 lines meet. This node represents a hypothetical ancestor of the lion, and this ancestor, well, some of its descendants became lions, but also some of its descendants became leopards. So from this tree, I know that lions and leopards are a group. They're sort of evolutionary cousins. If I follow it back farther, I can follow this branch back and I reach another node. This again is a hypothetical, theoretical ancestor here, and it left some descendants that evolved into some species that later became lions and leopards, and it left other descendants that became jaguars. So again, I can look at this as another group. These things are clearly related to each other. And this makes sense. The lion and leopards are the big cats of Africa. The jaguar is the big cat of South America, and the Africa and South America used to be touching. So that makes sense in terms of what we know about these species. And then just to finish this off, of course, over here we have the tiger and the snow leopard. If we bring these branches back, we find the node. They share an ancestor. That ancestor became tigers. That ancestor, some of the descendants became tigers, some became snow leopards. So this is another group. And again, that makes sense. Those are the Asiatic cats. And any tree that you follow back, eventually you'll get to the root of the tree down here, this ancestor that eventually had descendants that led to all these different animals. So we're going to do this. We give you the sequence data in a text file, and we encourage you to use a program that builds trees. And we talk you through using this program called Simple Phylogeny. We do this one because it's for free online, and it's easy to use, and it will build a tree in just a couple minutes. So when you put all this data that I told about into Simple Phylogeny, it's going to give you back a tree that looks like this. All right, let's see what's going on here. First off, I see some of the animals, some of the lemurs that are the ones from the study here, some more that are down here, and then I see these other species in groups that we put in the tree as well. So first off, I see down here Mbola, Razafi, Alicia, Bima, and Ihari. Well, that's that that group of 5 lemurs that we believe to be the furry-eared dwarf lemur. And as I trace all these branches back, sure enough, they meet at a node. They have an ancestor whose descendants became all of these different lemurs. And so we believe, yes, absolutely, that is a species. They are more closely related to each other than they are to anything else on this tree. We knew that going in, however. And the interesting part is up here, Malala Garafa Rudolfo. They also have a node where they all meet before it meets with any other organisms on this tree. So these animals share an ancestor whose descendants became them and no other lemurs. That is the best evidence we can have that these are a group. These are indeed their own species, and based on the morphological evidence we have, we believe that this is C. sibriae, the Sibrie's dwarf lemur. We can learn some more from this tree though, right? If we come down here, we find another node, the medius, the fat-tailed dwarf lemurs. If we trace their ancestors back, we meet at this node here. Now the interesting part here is that when you trace these two things back, you meet at another node. This tells us, at least based on the Site B data, the C. sibriae, the Sibrie's dwarf lemur, seems to be most closely related to C. medius, the fat-tailed dwarf lemur, and not C. crossley. So not only are these different species, but they don't even look to be that closely related. Not surprisingly, the C. major species all meet back here at a node, and sure enough, these things used to be we used to think they were one species, and sure enough, we follow them back. They share a node back here. So they are closely related, but again, they're different groups, so clearly different species. All of our dwarf lemurs meet at a node back here, which makes sense. They're dwarf lemurs. And then the one other thing on our tree, the gray mouse lemur, we knew this was different going in. We put this in as a frame of reference, and depending on the program you use, sometimes the programs require you to have what's called an outgroup, something that you know is more different just for technical reasons. So that's why that one is included. So again, this absolutely confirms C. sibriae is its own species. It's a species we have rediscovered that was thought to be extinct. But I just want to show you the range of C. sibriae today. 3 and perhaps 1 or 2 more small fragmented places they've been found. They are extremely endangered. They live in highly fragmented patches, and when animals live in fragmented patches rather than in a continuous range, that puts them at even more risk of extinction because their effective population size is then much smaller than what the actual total is. So C. sibriae is a species that we definitely have concern about. And that will really affect our conservation decisions because it lives in the range of other species of lemurs that we thought were that we had less concern about. So these patches specifically where they live, they are prime candidates for really intensive conservation. And again, they are considered critically endangered by the IUCN, and they're only found in those few fragmented forest patches. So to wrap everything up that we've done today, we use modern genetic techniques to analyze authentic data and rediscover a lost species of lemur. To do that, we use the tools of evolutionary biology to build species relationships. And once we understand those species relationships, we can use that and those genetic techniques to rethink how we interpret biodiversity and therefore approaches to conservation. So I really just love this lab because not only is it fun to look at lemurs, but it's a hands-on lab that directly deals with evolutionary evidence and conservation evidence. And those are 2 places in curriculum, whether you're teaching general biology, AP biology, AP environmental science. those are direct places where it's hard to get hands-on labs to do. But we really think that we present something here that's worthwhile doing, and puts a pipette in a kid's hand, which is a powerful thing to actually look at real data and analyze real data. So with that, there were just, when folks signed up, there were a few questions, and I realize I've gone a little long here, but we'll try to get through them quickly. So one question was, how close are the techniques used in this lab to ones actually used in the field? Well, that's one of the things that we really like about this lab, is that they're very similar. In fact, some of the tools that you can use in this lab are tools that people are using in the field. Absolutely, for sure, the morphological data that folks are looking at, that kids will look at in this lab, is the same morphological data that people looked at in the field. So that's the same, without the fun of actually measuring the live lemurs. The PCR RFLP is an absolute, authentic technique. It's not one that is necessarily regularly used for this purpose, but it is a way that we can get students using authentic techniques to look at data in the lab. And the RFLP, the polymorphism that we're looking at, is a real polymorphism between the species that we talked about. The sequence data analysis is absolutely, we're looking at the sequence data collected from these animals and using authentic tools to put that sequence data together. And again, the ecological data is, again, actual data collected from the field. Is there a way to do this lab remotely? So yeah, in today's world, we're not all in a laboratory. We understand that. So I'll just say we presented 4 activities here. 3 of them, the morphological analysis, the tree building activity, and the ecological data, all those are either paper or computer based. We put all our curriculum on it'll be on the product page for you to download. You can download that stuff for free. So while we encourage you to purchase the lab, nothing is going to stop you from downloading that the user guide and just doing the virtual activities or doing them remotely and skipping the hands-off portion of the lab. Yeah, we encourage you to do the hands-on portion of the lab. Again, putting a pipette in a student's hand and having them answer questions in that way, I believe, is really powerful. But the other resources are there available to you. What resources are available now, and when will the lab be released? So, as I mentioned, this is up here. This presentation is up here. It's going to live at this website going forward, so you can feel free to share this. Also down below in the description here are questions that you can give to students if you want students to follow along with this presentation. And then also these PowerPoint slides will be uploaded to that to the description sometime tomorrow probably. So those are all available. The lab, we were hoping to have it released by now. With the way the world is, some priorities got shifted around. We expect it to be released by early next year. So with that, I just really want to thank everyone who spent this hour with me. I'd like to thank our collaborators, Lydia and Marina, for spending the time in the chat and for graciously allowing us to use their data and to develop this activity together. It's been a wonderful collaboration, and we really think we've developed something that will be useful to teachers going forward. So on behalf of miniPCR Bio, on behalf of the Duke Lemur Center, thank you for joining us today. And with that, have a wonderful rest of your day.